So last week we talked a little bit about supervised learning in the context of linear and nonlinear models, and we gave a brief overview. Today I want to go a little bit deeper in detail in supervised learning in the context of classification and regression. So just to recap what we have been talking about, we already talked about response variables or labels, which are the outcomes that we want to predict. Labels can be categorical. For example, if we are trying to predict case control, responder, non-responder to a medication or treatment group. And the labels can also be continuous numerical values or floating point values, like optical density, patient age, or disease severity score. So if we are talking about categorical labels, if your data set, the outcome you want to predict is categorical, then we will use classification models or classifiers, like random forest classifier or logistic regression classifiers. If we are talking about continuous numerical targets, then we'll use regression models or regressors, linear regression or random forest regressor. So depending on the type of label you have, you will choose whether to use a classifier or a regressor. But before we get into more detail on the differences between these two types of models and how they are evaluated, how they're used, I wanted to talk a little bit about covariates, confounders, and batch effect. I know this course is more focused on machine learning, but if the study design and the data are bad, your machine learning model will fail. It will be terrible. So data and machine learning models, they walk together. Both need to be good in order to excel in a machine learning workflow. So I thought it was important to mention that. And in the Colab notebooks exercises for this week, we go a lot into detail on that. And we use real examples from publications in high impact journals. And we reproduce the results and we identify major issues related to those things. So in addition to the input features, the predictors, and the outcomes, we can also have covariates. So for example, if you are working with epigenetic data to try to predict the risk for a certain disease or the response to a medication that is interacting with the epigenetic profile of a patient, age and sex could definitely impact the epigenetic profiling of patients. So if you don't include those covariates, those features, additional features that are not exactly your predictors, but they impact somehow the association between your predictor and the outcome, or they impact the outcome in a certain way that complements your predictors, then you'll be missing some potential power or performance in your model because your model is not seeing the full picture. So it's very important to identify in your study what are potential covariates that you should include in addition to just collecting data. For example, in gut microbiome, diet and use of antibiotics and previous infections are key covariates to include. Epigenetic studies and genetic risk prediction studies, sex and age are other important covariates. There is another type of feature, another type of variable. There are confounding factors or confounders. They are variables that impact both the predictor and the outcome. And if they are not adjusted, then they can cause bias in your model. So if you have a confounding factor that is impacting, for example, your training data set that you're using to build your model, but that confounding factor is never taken into account or filtered out or regressed out from your data, and then you're gonna try to use your model on another data set that is not impacted by that confounding factor, then your model will underperform. You'll have a low accuracy. And within that area related to that as well, there's a batch effect, which is not exactly a confounding factor, but it's a type of technical variation that is not really true biology, that is caused by the way you design the experiment, the way you collect the data, the way you recruit patients. If you have a batch effect in your data, the models will learn those batch artifacts, those non-biological associations, instead of learning the actual biological signal. The model will always try to cheat and find the easiest way to predict the outcome. It will not have ethics and use biology to predict things. So you have to be mindful about that. So just in this example here on top, if you design your experiment poorly and you put all the cases on batch 1 and all the controls on batch 2, and then your model finds an association between a certain species here, a certain relative abundance that drives a higher probability of being a case versus a control, you don't know if that's true. It could be driving a higher probability of being batch 1 instead of batch 2, because there's a perfect match between batch ID and case, or batch ID and case control status. And this effect could be caused by the way you handle the samples, not actually by being a case versus a control. So you never know if you design the experiment like that. We don't know which one is the right assumption here. Which one is the right hypothesis? Is it the batch effect or the case status that is driving this differential abundance? So if you do a better design of your experiment, you put similar individuals, like individuals, case control matching, individuals that have similar features. The only thing that they're different is case and control. They belong to the same batch. Then you kind of eliminate that. You mitigate, you minimize, you don't eliminate, you minimize that batch effect, because now you can disentangle that signal and you can see if your model is able to differentiate cases and controls across all batches. One way that I usually personally do, a quick test that I do to see if there's still batch effect on my data, is to build a model where my outcome is the batch, just to see if my model is doing a good job in predicting the batch ID. Then it means that my data had a poor design and there's still a lot of batch effect in there. So it's a good sanity check to just make that toy model. That makes that just that sanity check model. Can we predict batch ID from our data? Then there's batch effect, and you need to either include that as a covariate in your model to regress out that effect, or you need to filter that out. There are different techniques to do that. Singular vector decomposition, for example, you can use that to filter it out, but this part here will not be covered in the course. We'll use easier ways, simpler methods to correct for those potential batch effects. So now that we talked about some potential data and study design issues, we also should talk about in more detail on how we evaluate our models, how the models are evaluated to be optimized and evaluated to be selected and to be improved and to be tested. We have talked about before on how models are fit, how they're trained. We start with our training data. If it's supervised learning, we have our input features, X. We have our ground truth outcomes, Y. And then we use a machine learning algorithm to build our model. And building our model consists of starting with random parameters, making a prediction, or Y hat here, And then we use a loss function to tell how wrong our model is. We use that loss value on our gradient descent optimization algorithm to tell the model how cold, how warm it is, which direction it should update the weights to then improve, right? That cycle keeps going until the model converges, reaches a solution that reaches a parameter set that doesn't improve anymore. But in the context of classification versus regression, we need to take into account what type of loss function is the most suitable for the type of label you have, or depending on the amount of data imbalance you have as well. One of the most typical loss functions utilized in regression problems is the mean square error, which is a very simple calculation you take in linear regression models, or random forest regressors or XGBoost regressors. All the regressor models, they typically use some sort of loss function that is similar to mean square error, where you start with your y-hat, your predicted label, your ground truth label coming from your training data, you subtract them, you square them, and then you calculate the mean. And that value will be proportional on how wrong your model is. So you want to minimize that value throughout the training. So larger errors mean that they'll be penalized more because the scale of the mean square error will increase. And common examples of applications include if you are predicting age, body mass index, blood pressure, those are continuous values. So if you build a model to predict those, you need to use that type of loss function. And sorry, I was covering the equation here. I hope you can see it now. Yeah. In the case of data sets that have machine learning tasks that have binary or categorical outcomes, then the model process the outputs slightly different. Instead of your y-hat being a continuous value that can go from minus infinite to plus infinite, your y-hat will be converted into a probability p, which is the probability of the class or the label being true. So usually the true label is the least abundant in the data set. For example, in a typical observational study, most of the people will be healthy. So the people that have the disease are the true label. So you assign 1 to them, right? So these y-hats are converted into probabilities, and then to calculate the binary cross entropy loss or log loss, you simply, whenever you have a ground truth label that is 1, you calculate minus log of that predicted probability. Whenever you had a true class that is 1, you just calculate 1 minus that probability, and you do the minus log of that. So that BCE loss is used for classification problems with categorical targets. The model outputs a probability, and then the BCE checks whether a high probability was assigned to the correct class or not, or how far that confidence or that probability is from the true class. So then confidently wrong predictions, if the model has a lot of confidence that that prediction is right, but it's wrong, then it will receive a very large penalty because of that log element. It helps the model to learn faster and then correct those bad predictions more aggressively versus using the MSE. That MSE does more like a linear type, gradual type of correction.